What I will be talking to you is on the basic aspects of epidemiology and some concepts that are related to epidemiology. And as a matter of fact, in the subsequent sessions, each of these concepts as well as definitions will get expanded with some examples as well as some real life scenarios that will be useful to you to further make use of these concepts in your day-to-day work. Broad objectives of this session will be to define formally what is epidemiology, and then also talk about what epidemiology is used for, what are the different uses of epidemiology, and along with the uses, we will be also describing key concepts that are needed to make sure how epidemiology works, and therefore in the VBD context, you can apply them in a real life in your own day-to-day work. Epidemiology is a core science, and we call this the diagnostic arm of public health. The reason why we call it diagnostic arm of public health is because as a science, epidemiology is used to detect, to diagnose problems in the population so that action can be taken through various methods, including organized programs and sometimes as a response to emerging problems. As a diagnostic arm of the public health, epidemiology is also helpful to make sure we know what we are doing is right, whether things can be improved, whether things needs to be changed, if at all we need to do something about the way we are implementing our programs. So basically, it's a core science, and it has gained its prominence now with the COVID time. Let's look at the formal definition of epidemiology. It is defined as the study of distribution and determinants of health-related states or events in specified populations, and the application of this study to the control of health problems. That's the definition. Basically, there are three things that are very important. One, the distribution of health and health-related states or events. Second, the determinants of those events, why they are happening. Third is in terms of using the knowledge to make sure we do something that is necessary to reduce or to control or to eliminate that particular problem. So the epidemiology is usually people talk about various types of epidemiology. Epidemiology, the science itself, is application oriented. Epidemiologists also do various other things apart from distribution, determinants, and applying science to make sure health problems are tackled properly. Epidemiologists also play a major role in conducting research and make sure this is reported to relevant stakeholders so that the research is taken to its next stages, including the core principle for which the research is done in terms of protecting and promoting health of the populations. Epidemiologists, as some of us are at the National Center of Epidemiology and elsewhere, they are also involved in teaching and consulting and majorly involved in also public health administration. So epidemiology as a science, as a core science, has this broad areas of application apart from what we initially stated as studying distribution, determinants, and application of knowledge in public health programs. As compared to a clinician, the role of an epidemiologist is to deal with population as a whole. And as compared to a clinician, an epidemiologist makes sure he frames the right kind of question to diagnose the problem in the population. A physician may check the patient for identifying some signs and symptoms and also detecting any problem through examinations, whereas an epidemiologist will investigate the health problem at the population level. The clinician may clinch a diagnosis, whereas an epidemiologist may come to a conclusion about why the problem is occurring and what can be done about it. The clinician may give a prescription, whereas an epidemiologist may propose what can be done about it. The clinician reviews or makes sure there is a follow-up of the prescription that was given, and also the prognosis is measured over a period of time, whereas an epidemiologist makes sure he measures how well the program that is implementing the interventions are functioning at the field level. So this is the major difference between a doctor or a clinician or a physician versus an epidemiologist whose primary function is to function at the level of a population. What are the different components of epidemiology? We learned that as a definition, epidemiology is dealing with three things. One, distribution of the health problem or health related events. Second, determinants of those health problems. Third, make sure the above two contributes to applying the results in the field. Therefore, the components of epidemiology includes measuring how much is the problem, how big is the problem, and also checks who is affected, where they are more affected, and what time point in what point in time they are mostly affected, and is there any clues can be drawn about these three characteristics in terms of time, place, and person. Therefore, we have an idea. That idea or information or pattern can be used to make sure we have a set of questions that can be answered in a systematic manner. The systematic manner, when I use the word systematic manner, it refers to making sure the ideas or patterns or the clues, in a technical term, they are also called hypotheses, can be examined through the third use of epidemiology to make sure we identify why the particular problem does exist in a particular place or in a particular point in time or among a group of people. The why question is answered through testing of hypothesis. That's the third component of epidemiology, an important component. We'll now look at uses of epidemiology. I have listed here 6 important uses. There are many uses of epidemiology, and I have taken the liberty to put a concise list which talks about precisely what epidemiology is all about. The first one that you see is about examining causation. Epidemiologists per se, epidemiology as a science per se, the core is to examine cause and effect, to examine exposure and outcome, to examine risk factor and disease, to examine why a person who is in good shape, good health, becomes ill, or why he is suffering from a particular disease. And as a logical science, this is a question that we always ask, connecting X with Y, connecting a particular factor with that of the outcome. Therefore, the first use is about examining causation. The second use about studying natural history. Third is about the health status in the population. And fourth is about determinants, and also look at relatively which determinant is important and which of the outcomes are more importantly happening in a particular given context. And fifth, make sure when you apply these principles in the field from the findings, you evaluate how they are working and whether they can bring down the burden or a particular factor. Sixth is to make sure you identify risk factors. Let's look at one by one. This diagram that you see on the screen is an oversimplification of how we examine causation in epidemiology. What I have given here is to answer the question why a particular individual or a person having a good health to begin with suddenly is down with or is suffering from ill health. The question is why. So why a good health individual becomes ill or diseased? This question is very important, and that's a primary question that always we deal with in real life. This causation could be about identifying things that can be fixed by us from program, by physician, or by interventions like vaccines, for example. Interventions that can take care of the causes at the broader level in the community. It could include various other things that we cannot fix. For example, genetic factors. It can include things that has to be taken care of at the global level. For example, environmental factors. It could be related to the behavior of the individual in terms of lifestyle risk factors. It could be anything that makes a good health person ill health. So Examining causation is the focus or central theme of epidemiology. I'm sure all of you in the field, you also ask this question, why somebody who was fever free until yesterday, today he is having fever? Then we start asking the question, did he visit some place? Is there a problem of, breeding? We ask a number of questions to examine causation. Without knowing, we are applying epidemiological principles in the field. Examining causation is the central theme. Second important aspect of epidemiology, which is very well recognized and used, is in terms of studying the processes that are involved in between good health and ill health. Processes is in terms of a person who is all right, suddenly within his body, without expressing the physical signs outside, he has some internally the disease process is set in. And then what we see is a clinical disease, which means he has signs or symptoms. For example, fever or rashes or diarrhea or any of the physical signs such as, for example, the blackening of the skin, things like that. That is a clinical disease. And then ultimately the patient or the individual may recover or die of that particular illness. This is called natural history. Natural history as such is an important piece which helps all of us to make sure we understand which steps can be useful to make sure we can intervene, or we should have screened, or we should take care of the individual with the suffering that a person has. To understand natural history, what we call as a triad or the three things, epidemiological triad, is very important. Natural history of a particular infection or a disease is an interplay between the individual, which is referred to here as host, and that of the agent, which could be any infectious or non-infectious agent. The third is in terms of environment, the place or the situation or the setting or the context in which that person lives. And the fourth one, which is in the middle, is what is very important to all of us who are dealing with vector-borne diseases, which is an intermediary that can come in between the three. So host has number of factors. Host is healthy because the host has strong immunity. Host has a good gene, therefore the genetic disorders is not coming. The host has good nutritional status. The host is a good host. That determines whether the host is in good health or bad health. Agent, as I said, could be infectious or non-infectious. The infectious are listed here as biological, and non-infectious could include physical agents or chemicals or things that cannot be quantified easily, psychological or social agents. Environment, when we say environment, it refers to not only the climate and things like that, it could include the water, sanitation, hygiene, air, and political climate, also socio-cultural factors. The environment could be anything that determines the increase or decrease action of an agent, and then increases or decreases the contact between the agent and the host, and then enables an environment where good health person can become ill health. Let's come back to the natural history again with some more details. If you look at on the left hand side and then we move to the right hand side, the typical process that goes as a result of natural history progressing from infection to disease. We are taking an example of infection because we are dealing with vector-borne diseases. So on the left hand side, you see stage of susceptibility, which means the word susceptible, I think it was mentioned by the director MD-NHM also. He also used another word called vulnerable. So susceptibility or vulnerability are the same, which means the person is at risk of getting infected. That's the meaning of the word susceptible or vulnerable. There are other words that people use, including, for example, whether the person is at risk or immune or not immune. There are different words people use. Basically, at that stage, when exposure happens, when I say exposure, the exposure could be due to the biological agents that we mentioned, and also it could be due to physical agents. It could be due to other agents that we mentioned, social and psychological. When the exposure happens, the person gets that particular exposure inside the body or inside the system, and then that induces some changes, damage, and the person is affected because of that particular infection or the agent. That sets in motion a process called pathological process, which means he or she was in good health. That infection or the agent has induced an effect by which the good health starts deteriorating, which means internally some problem occurs. It creates a damage. If the damage is not expressed as a disease or a symptom or a sign, then it is called stage of preclinical, which means it is subclinical. It is not still expressed, but the damage is already occurring. For example, for some of the infectious diseases, fever may not be the first sign. The patient or the individual may be harboring it. It can be only detected by testing blood or some body fluid. So the second step is expression of clinical symptoms. The patient, as a result of the infection or the agent coming in contact with the body, getting exposed, expresses some disease. It could be like what I said, fever or rash or vomiting or headache or retroorbital, the eye pain and things like that. That is called disease. The period between the time of infection to the expression of the disease is called incubation period, which is very important in the context of infectious diseases. Then comes the usual stages of progression. One, you will be able to now clinch the diagnosis, saying that this diagnosis is dengue hemorrhagic fever, for example. And then as a result of the disease and its consequences, depending on the management and also various other factors, including the host factors, the person may recover or get some residual effect of the disease or may die. So this is called natural history, a full spectrum starting from disease-free status to an extreme of either dying or suffering or recovering. This is called natural history of communicable diseases. Why are we interested in this epidemiological triad and natural history? This helps us in making sure our interventions, we need to know when to do. Our interventions, we need to know when it will be working. Our interventions, we need to know when it will work and when it will not work. If at all we have to diagnose a patient very early stage, we need to know at what stage we can diagnose. And also, we need to measure the response to our treatment or management or whatever we do. So, natural history is very important. There is another concept that is also very important called chain of infection. Chain, it's called chain because it's a linked process. It starts as there is a pool of infection. From the pool, the agent pops out and then gets entry into the human body or the individual's body and then triggers the process inside the susceptible host. The chain is a chain between reservoir, root, and the host. When I say reservoir, it could be anything. And then from the reservoir, through some route, it exits. It comes out. That's called portal of exit. And through some entry point, it enters into the host. That is called portal of entry. Between the two, it's how it gets out of it and then gets into it. It's called mode of transmission. It could be direct. It could be indirect. It could be many other methods. For example, in the context of COVID, it's due to droplet mode. And from one person to another person, it travels between through a medium, which is usually droplet. Okay. With this knowledge of natural history and that of the triad and the chain of transmission, what we do is we try to interrupt the chain of transmission. We can, for example, I'm just going back to the epidemiological triad. So, for example, we can intervene at the state between the agent and host, we can attack so that the agent doesn't come in contact with host, or make sure the host is protected by a drug or a vaccine. We can also reduce the agent in the environment, and we can also make sure the environment is free of the agent. For example, some of the things that we do, remove the stagnated waters, is to make sure the water, the good water, quality water, doesn't allow the breeding of larva. So we make sure with this knowledge, we intervene, we interrupt, and then make sure we either eliminate or control the source. The source could be human by isolating the patient or treating the patient. The source could be environment, or reservoir could be environment, where we make sure we contaminate, decontaminate, or make sure we drain the stagnant water. If it is animal, we isolate, treat, and cull. With reference to mode of transmission, we interrupt by using various methods by the knowledge that we discussed. How does the transmission happen? Is it direct or indirect? Is it through some vehicle? If we know that knowledge, then we make sure we cut that chain of transmission that happens between the between the agent and the host and the environment. For example, in the vector-borne disease context, we eliminate the vector, we use insect repellents, and we use a lot of methods to protect human from getting bitten, and so that the transmission doesn't occur. Last but very important, we also need to cut the chain, or control the chain, or interrupt the chain by making sure the host is protected through by giving drugs, which is called chemoprophylaxis, or by giving vaccines, which is vaccination or immunization, or the ultimate is avoid exposures to that environment, in that environment where such vectors could be running around. The third use of epidemiology is in terms of describing the health status of the population. What do we mean by that? We mean who is affected, to what extent they are affected, which time point in they are affected, and where there is most affected. We describe the health status of the health status of the problem in the population, and make sure we have a good understanding of who, where, and when. For example, in the given diagram, we are talking about age and gender specific burden of malaria in a given population by looking at age structure and gender structure, and then looking at number of population in those age and gender structure, and we have number of cases for each of these age and gender structure, and we calculate what is called attack rate, or you will learn more about it. We legitimately use the information available with us to make sure we have a good understanding of who is affected, where they are affected, and to also understand at what time point in time this is more. The fourth important use of epidemiology is in terms of determining among the causes. Illness could be brought on by different causes, different risk factors. Among the different risk factors or causes, understanding how important is one versus other gives us an understanding where should I invest first. For example, the National Health Mission needs to invest on the priority health problem. How do we identify priority health problem? For example, in the given slide, we have given tuberculosis, we have given measles, we have given malaria. So the second column is called DALYs, which represents the disability adjusted life years for each of these problems. The third column gives us mortality that is attributable to each of these three conditions. The question is, where do I invest my money to make sure I keep track of these three diseases? Which one is more important? Then I look at these information about which one is burdensome in terms of its disease or in terms of its consequences, and then I make sure I invest on it and keep the population healthy. So fourth important use of epidemiology is to make sure we have a good characterization of relative importance of what is important and what is what needs attention from the authorities. The fifth important use of epidemiology is to make sure we know what we have done is correct, or what we have done requires tweaking, we also would like to know whether the intervention that we have applied is yielding desired effects or not. Therefore, by making sure whether our interventions are going in the right direction or not, whether it needs some updation or tweaking or reconsideration in terms of either the methodology by itself or changing the methodology of employing that particular intervention. We need to evaluate our interventions. That's an important component in epidemiology. We have methods by which we systematically look at how well are we doing with reference to a particular health program. So this is an important use of the epidemiological science. Sixth and the last important component of epidemiology, the use of epidemiology, is in terms of making sure we have evidence. I wish you mark this word evidence. We have an evidence which is convincing, which has its own basis from science, to make sure we have identified typical sections, specific sections who are at greatest risk through a particular risk factor that results in ill health. For example, the table shows the encephalopathy syndrome, which is causing every summer children dying in Muzaffarpur in Bihar. So, for example, this particular table shows that if you look at the yellow marked portions, among those who died of that particular encephalopathy syndrome versus those children who did not die, we have made a comparison and we have figured out that 65% of those who died have taken that particular fruit lychee as compared to 23% among those who are alive, which means 65% as compared to 23%, there is a big difference in terms of that particular risk factor called eating lychee 24 hours prior to the disease process has set in, which indicates that those who have taken lychee were almost 10 times highly likely to have died as compared to those who did not take lychee, which means it's a powerful risk factor. We need to do something about lychee as compared to, for example, there are other risk factors that are listed over there, whether they had visited a particular area for work, whether they have their parents have visited, they had meals before evening, previous night, whether they belong to a particular socioeconomic strata, whether they have washed completely the fruits and vegetables before they were ready for eating or cooking. So relative importance of specific causes is identified through epidemiology. So that's another important component. I would like now your attention to talk about three broad components, approaches in epidemiology. One is called descriptive epidemiology, the other is called analytical epidemiology, the third is interventional or experimental epidemiology. Descriptive epidemiology deals with what we learned from the definition, the distribution. And I also mentioned about quantifying the problem and then talk about who is affected, where they are affected, when they are affected. That exactly is descriptive epidemiology. So epidemiological approach involves distribution of the problem in time, place, and person. So that's called descriptive epidemiology. We also mentioned that descriptive epidemiology helps us in identifying a pattern or a clue, or in a technical term, it's called hypothesis, so that we have a research question that can be taken to the next stage. The second approach is called analytical epidemiology. Typically, the word analytical refers to use of a comparison group to make sure we test the research question or a clue or the hypothesis, whether it is true or not, by comparing, for example, diseased versus non-diseased or exposed versus unexposed. And then identify the determinants, which is the second purpose of epidemiology. The third approach is to make sure we do experiments. In epidemiology, the experiments could be at the clinical level or could be at the population level to answer which interventions are effective in making sure a particular outcome comes down or works much better than the other outcome. That's the third approach of epidemiology. One of the bases of epidemiology is to apply the principle of count, divide, and compare. This is very important because epidemiology, as we said, deals with population. So we need to have denominators. And therefore, we use the principle of counting the diseased or number of people affected with a particular problem, and then make sure we divide with an appropriate denominator and then make comparisons. For example, the first step count the number of cases here shows number of malaria cases in city A versus city B. City A has 58 cases, city B has 35 cases. So you may think that city A has more number of cases as compared to city B, but hold on. Make sure you divide using the appropriate population belonging to city A and city B. And interestingly, the next table gives information for city A for 1 year, and for city B, information is given for 2 years. So for city A, 58 cases were reported in 1 year, 2004, from a population of 25,000, whereas city B reported 35 cases for 2-year period from a population of 7,000 there. Therefore, having found the denominator, our job must be to divide. When we say divide, we divide the numerator, which is the number of cases reported, by the total population for that particular city or a setting or a place. So what we find here is city A has 58 per 25,000 per year, and city B has 35 per 7,000 for 2 years. The next step is crucial. Dividing alone doesn't help. We have to make a comparison. So when we compare the indicators between city A and city B per year, city A has 232 per lakh and city B has 250 per lakh per year. Now we have converted for city B, since we had 2-year information, we have converted that into 1-year information and calculated city A and city B malaria rates, malaria frequency into per year, per 100,000 per year. Here you will see, surprisingly, earlier when you saw the number of cases, city A had the highest number of cases, city B had the less number of cases. Now, when you use the population denominator, actually city B is at higher risk and having higher burden of disease, malaria, as compared to city A. This is the basis of epidemiology. By using denominators, we make sure we divide with appropriate denominator. When I say denominator, we also use the word susceptible in the natural history diagram. I use the word at risk. So the denominator should be population at risk, appropriate population. And then we make sure we compare the indicators between the two settings or two scenarios, and then make appropriate inferences about that particular problem. This is a very basis of epidemiology. And to summarize the basic principles of descriptive epidemiology, in the descriptive epidemiology, we analyze time, place, person and use all the CDC, count, divide, compare, to make sure we look at it by time, we look at it by place, we look at it by a person. In all of these, for example, in time also, you can look at, for example, in even a particular year, you can allocate the number of population for that year. Appropriate denominator is used. If it is place, you give total population belonging to that particular place. It could be a house, it could be a street, it could be a village, it could be a tehsil, or it could be a subdivision, or whatever. Appropriate population is used. And finally, we use person information for particular age or gender or socioeconomic strata, whatever, and make sure you calculate descriptive epidemiology of disease burden by person. And we then use the information that is derived from descriptive epidemiology to the next stage of analytical epidemiology. And as I said, in analytical epidemiology, we specifically use comparison groups to make sure we test them out and rule out the role of making the inference stronger. And we also apply statistical principles while doing so. For example, when we say comparison group, we can use, for example, exposed versus unexposed, in which case we follow up a group of people who are exposed, we follow up a group of people who are unexposed, follow them up over a period of time, and see whether disease frequency occurs more frequently in the exposed group as compared to unexposed group. which is called a cohort study. So we can do a cohort study, which is an analytical study to examine the hypothesis derived from descriptive epidemiology. Second, we may compare diseased and non-diseased. A particular area has people who are affected with a particular problem, and we then compare another individual within the same village or a setting who is not affected, and then make valid comparisons in terms of whether both of them have a particular exposure factor associated with. And if one exposure factor is highly associated with a diseased individual as compared to a non-diseased individual, then we clinch the particular diagnosis about which problem is affecting more often the cases as compared to controls. And this is another way of testing the hypothesis. Cross-sectional studies, which are very popularly known as surveys, can also be used to make legitimate comparisons. For example, slum population versus population living in main area, and then make some comparison about some of these, say, awareness about vector breeding habitats, things like that. And the third, which we already mentioned, you can use experimental epidemiology to draw valid conclusions about which of the treatment strategies or intervention strategies are effectively working in reducing or favoring a particular outcome. So these are the main approaches that we use in epidemiology. So thank you very